Hello everyone. In this video I'm going to be talking about the Operon or lactose operon as it's sometimes referred to. And this is a topic only really studied in A-level biology, so it's really for the Key Stage 5 students that I'm aiming this particular video at. Now, typically before studying the Operon, students are taught about genetic control of protein production in eukaryotic cells. And you may have gone into detail about gene silencing with short interfering RNA, or how cells are regulated pre and post transcription and translation. But in this video, I intend to just focus on bacteria, or specifically E. coli and some other enteric bacteria, and how they are able to control how their genes operate, and how they are able to control the proteins that are produced, by looking at how the Operon functions. Now, before we speak of exactly what the Operon is and what it does, it's essential to give just some background information to this. Now, what Jacob and Monod did, because we would usually refer to the Jacob-Monod model, is they looked at how bacteria respond to changes in their environment. Now, they need to respond to changes in their environment very, very quickly. And I asked my students these two questions. If they find new nutrients, such as lactose, they need to make enzymes to deal with it. And then I asked them why and how. Now, the why would be that this new nutrient, such as lactose, could be used as a respiratory substrate. The how would be to turn genes on to code for enzymes to be able to deal with it. Very simple. I then asked, after the lactose is broken down or gone, they would need to stop producing the enzyme for it. Again, why and how? The why would be because otherwise you'd metabolize too much and it would be wasteful, and the how would be to turn those said genes off. Now, this is some of the work that Jacob and Monod looked into, and basically they said these three things: that the rate at which an enzyme is made in a bacteria depends upon the environment it's in. E. coli is able to metabolize lactose as well as glucose as a respiratory substrate, and to do so it needs two enzymes: lactose permease to get the lactose into the cell, and beta-galactosidase to break the lactose sugar into glucose and galactose once it is inside the cell. And finally, E. coli grown in a culture medium without lactose will have very little quantities of these enzymes. If then placed in a medium of lactose with subsequent sugar added, then the rate of synthesis of these enzymes then increases. So what they basically concluded was that lactose is a trigger or inducer. And that's one thing we're just going to make a note of at the top. So in this whole thing, lactose is our inducer. And we'll come back to that word in a moment. So lactose is our inducer. So let's think about what the Operon actually is. An operon is a strand of DNA in the bacteria's DNA, and it composes of genes with related functions. So we're going to draw a little bit below here. In fact, we'll do this in black. We'll draw what the operon looks like. So here we have a length of DNA. And in it, we've got genes of related functions. That's by definition what an operon is. We have a starting point here, a region. We'll just shade this part in red. We'll just do it in different colours. This is known as the I region, or the regulatory gene. It's not technically part of the operon, but it acts to produce what's called the repressor protein. So we've got I, and this is the regulatory gene. We've got two regions here, and we'll just shade those in just so we're clear on what the two different things are. So we've got a green one there, put a pink one in here. Now, these two regions are known as the P and the O region, and collectively these two are known as the control sites. Now the P, if we just take these separately, the P region is known as the promoter. And the promoter is essentially the RNA polymerase binding site. It's a starting point for transcription of the genes in an operon. So that area P that I've labelled is where RNA polymerase is going to bind. And you might remember that RNA polymerase, when it binds to a section of DNA, initiates transcription. The O stands for operator. Now that switches structural genes on and off, and we're going to just come to the structural genes now. So you've got the P, the promoter region, the O, which is the operator, and the operator basically switches these two parts on and off. So we'll just colour these two sections in here. One just very rough. And two. Now these regions we're going to call Z and Y. And these regions here represent, as I've just said, our structural genes. Now Z and Y are the two structural genes that I've referred to just at the beginning of this video. So one of them, Z in fact, is the gene that codes for beta-galactosidase. And if you remember, this was the enzyme that breaks down lactose. So once it's into the cell. But I did say we needed one particular enzyme to bring that lactose into the cell in the first place, and that's what Y is. Y is the gene that codes for lactose permease. So here's our basic setup. The operon, a strand of DNA in the bacteria's DNA, composing of genes with related functions. And you can see that all of these sections, the I, P, O, Z, Y, they're all part of this same thing. We've got an operator which switches structural genes on and off, structural genes being Z and Y, genes coding for two enzymes that allow the bacteria to bring lactose into the cell and metabolize it. And we've got a promoter region, which is where RNA polymerase would normally bind to start the transcription of structural genes Z and Y. And then on the end, we've got this I region, this regulatory gene region that's not part of the operon but acts to produce what's called repressor protein. Okay, so we've said fundamentally that bacteria need to respond quickly to changes in their environment. Now, if they're not used to being in a culture medium with lactose in, they would have very little quantities of the enzymes that would essentially break down and metabolize lactose. So let's think about when lactose is absent, what is actually happening, how this bacteria is enforcing this element of genetic control. So we're just going to shrink the screen a little bit here just to make some more room, and we'll move this up. So let's think about when lactose is absent. So lactose is absent. What is actually happening with this operon? Well, the first thing is that the regulatory gene is expressed. So that I region right here, we're actually going to express that gene, and it's going to produce what's called the repressor protein. Now this protein has two binding sites. One is for lactose, but one is also for the operator region. So essentially what we have, if we were to draw very, very quickly, I'm not going to label it all, but if we just, I was to draw a mini and I'll just highlight the regulatory gene region there. So a quick sketch of the operon. What we essentially have is a piece of messenger RNA forming, because if we're going to express that region labelled I, we're ultimately going to get messenger RNA of that gene being produced. That's going to be read or translated at a ribosome, we're going to end up ultimately producing, if I just draw this as a little purple blob here, repressor protein. So what we've basically had is the red region, the I, or regulatory gene region, produce a piece of mRNA, that's this little blue strand. We've got it being translated at a ribosome, which I've just labelled here as the shaded black part there. We're producing this purple dot, which is a repressor protein, and that repressor protein is going to bind to this part here, this operator region. Now if we think, if we're going to bind that protein to the operator region, what we'd actually get is it covering a part of the promoter region. So when this repressor protein, if I just put RP next to it, when this repressor protein goes to bind to the operator region, it actually blocks a little bit of this promoter region here. And when it covers that little bit of the promoter region, you can't get RNA polymerase binding to it. So you can't get transcription of the structural genes. So without transcription of Z and Y, we're not going to produce any of the two enzymes that we need. We're not going to produce any lactose permease, we're not going to get any beta-galactosidase, because we're not getting transcription of Z and Y, because the RNA polymerase cannot physically bind to that promoter region, because our repressor protein that's sitting on the operator part is covering a part of it. So we're not ultimately going to produce any enzymes to be able to break down lactose. Now let's think about how we can reverse this. So what happens when that bacteria is in a medium, a culture medium, where lactose is present or lactose has been added? How does the bacteria then cope with that, the presence of that substrate? And how is it able to then use it and break it down to become a respiratory substrate, to be able to get energy from it? So let's have a think. Let's look at now when that lactose, or when the inducer is present. So lactose, or lactose inducer molecules, by inducer we mean it can induce some kind of change, bring about some change. That lactose acts as an inducer molecule, and what it does, it binds to the repressor protein, changing its shape. So again, we'll just very, very quickly just draw just two particular sections this time on here. I'll have the promoter region and we'll have the operator region here. And what we had was this repressor protein. I'll just draw it in crosshatch there. We can see it was sitting on the operator region here, but covering a part of our promoter region, so we couldn't have RNA polymerase binding. When we introduce lactose, or these lactose molecules, and if we were to say that these blue dots here is our lactose, when lactose is now present, then what happens is that it binds to this repressor protein. It changes its shape, so it can no longer bind to the operator region. And the repressor molecule that is attached to that operator region disassociates, or comes off, if you like. So if we imagine this purple dot now coming off that operator region, you can see that we now have the promoter and operator region fully exposed. RNA polymerase can now bind to that promoter region. So if we put in some, just for the sake of argument, if I just draw this as a red square here, and if we said this is our RNA polymerase, we can now have that bind to our promoter region. because the repressor protein has essentially disassociated and come off. So what that means is subsequently we can have gene Z and Y being transcribed. The enzymes needed to metabolize lactose can be produced. And what that means ultimately is that we can enable the bacteria to produce sugars which can be used for respiration. So we have this lactose inducer, it binds to the repressor protein. This inducer-repressor complex can no longer bind to the operator. The repressor protein leaves the operator, and what we get is RNA polymerase now successfully being able to be bound to the promoter region. We get transcription of genes Z and Y, so we could, and I could draw it out again, but there's no need to, but I could draw messenger RNA of both the beta-galactosidase and lactose permease genes. They get translated at ribosomes, and we produce these particular enzymes. And now the bacteria is able to break down lactose and use it as a respiratory substrate. So that, in a nutshell, is the Operon. It's a strand of DNA in the bacteria's DNA, composing of multiple genes with related functions designed to ultimately control protein production. In the presence, or rather, in the absence of lactose, the bacteria doesn't produce major quantities of these enzymes because it doesn't need to. And then when lactose is present, this trigger or inducer enables or allows the bacteria to then start synthesizing the enzymes to deal with it. And this is just one way that the bacteria responds to changes in its environment. Okay, hope all that helps.